Hi everyone, my name is Sanjana. I hope you guys are doing well. So today let's discuss about DNA replication. So, what's DNA? DNA is a condensed form of chromatin that wraps itself twice across the circle structure called histone, causing a nucleosome. So, what is DNA replication and where does it really occur? So, it occurs in the S phase of the cell cycle. So, we know that a cell cycle has four phases: the G1 phase, that is the growth phase, the S phase where the DNA synthesis occurs, and the G2 phase where the growth and preparation for further mitosis occurs, right? And what's the final step? The M phase, that is the mitosis where the actual cell division occurs. Now, why did I get this here? Because this cell cycle is really important for us to remember in terms of where does the DNA synthesis really occur. Okay, so G1 phase, S phase, G2 phase, M phase. So now I'm circling the S phase so that you all remember that this is the area where DNA synthesis occurs. Okay, yeah. Next, let's go to the list of enzymes. So, the first glance, you might be like, oh my god, all these enzymes, what is this enzyme? So, but I'll try to make it simpler for you. So, here is an zoomed form of a DNA. So, you can also see that I've written DNA is anti-parallel. What does it really mean? So, I have drawn a image of two parallel lines, one from 5 to 3, one from 3 to 5. So, why have I really brought this here? We have to remember that DNA replication is anti-parallel. That means DNA replication always occurs from 5 to 3 direction. Always. Okay? Either it can be continuous, either it can be discontinuous. Discontinuous is when it occurs from 3 to 5 direction, because continuous is always from 5 to 3 direction. Okay, let's move on to the next thing. So here I have again brought back this zoomed DNA image, and I have further tried to make it much simpler. Please excuse my drawing, but yeah. So what is this thing on the right? So the right part is a small, easier form or easier part of the DNA on the left. So you can see that this looks very similar to this on the left, right? I'm cutting this on the left, trying to show you which part have I really zoomed. Okay, this area I have zoomed on the right. Okay, and when you see this right structure, you suddenly feel like it looks like a fork, right? Why does it look like a fork? Because that's basically meant to make it easier. It's called the replication fork. Okay, so the replication fork is the Y-shaped region where the DNA replication occurs. So we select an origin of replication, a point where a replication will start, and then we zip through. We zip through that origin of replication, forming a fork structure, and that enzyme that helps in the zipping is called the helicase. So I'm drawing the helicase now. The helicase enzyme that zips and attaches. Okay, original replication is found, replication fork is formed, and this replication is formed by the helicase. Now further, DNA, both the two strands of the DNA are separated now. Now what next? We bring about an enzyme called topoisomerase. So what does topoisomerase do? It prevents the supercoiling of the structures. We know that DNA is a condensed form. It's a supercoiled structure in the nucleus, in eukaryotes and cytoplasm in the prokaryotes. But in order to prevent the further supercoiling, we need a structure enzyme known as topoisomerase. So I'm changing the color so that it doesn't confuse you between helicase and topoisomerase. So helicase is yellow, which zips it and binds, okay, at the replication fork, and topoisomerase that causes the break and removes the supercoiling of the DNA. Fine. What next? Next, I'm going to talk about the enzyme known as single-stranded binding proteins. So what are these single-stranded binding proteins? Single-stranded binding proteins are these small enzymes that bind to each single strand that we have broken down. So why do we need these proteins? They prevent the re-annealing or the degradation of these single strands. So we know that both the strands of the DNA on the left, we can see that they are together, and we have separated it on the right. So in order to keep it that way, we need the single binding protein. Okay, what next? What enzyme comes next? Next will be our main character in the DNA replication. Okay, that's called the DNA polymerase. Now I have drawn 3 and 5. And why have I put the DNA polymerase in the 5 end? Because I previously said that DNA replication always occurs from 5 to 3 direction. Got it? Fine. Now for this DNA replication to occur by the DNA polymerase 3, we also need another enzyme known as primase. Now what is this primase? So primase is an enzyme that makes RNA primers for the DNA polymerase 3 to begin its replication. So you see RNA primers formed, and now the DNA replication will occur from 5 to 3 end. 5 to 3 end. Fine. Yeah. So I'm going to take another color. Excuse me. Yeah. So I think I found my color. Yeah. So I'm drawing this, and you can see that it is continuous, right? There are no breaks in my replication. It's continuous. I'm trying to make it as continuous as possible. Excuse me for my mistakes in my art. Yeah. So from 5 to 3 end, the replication always occurs continuously by the DNA polymerase. It moves. It keeps moving from 5 to 3 end and causing a continuous DNA replication. Now what about the 3 end? What happens there? Okay, as I already mentioned, DNA replication always has to occur from 5 to 3 end. So we go the opposite direction, right? DNA replication is what? Anti-parallel, right? So one is going from left to right, and the other one will go from right to left. So what are these small circles that I'm drawing? What do you think it is? So it is again the primase. So this primase forms small RNA primers for DNA polymerase to act, DNA polymerase 3. But why is it discontinuous? Like we previously discussed, DNA replication can be continuous and discontinuous. Now these discontinuous fragments are known as Okazaki fragments. So DNA polymerase binds to the RNA primer and forms discontinuous strands. Now these discontinuous strands are bound by an enzyme known as DNA ligase. So that forms a phosphodiesterase bond between all the small Okazaki fragments. Fine. Now we have so many enzymes attached, and our replication is almost done. What do we do next? Now we remove these primers. How do we remove it? By enzyme known as exonuclease. Okay, exonuclease functions are generally done by the DNA polymerase itself. Just that from 5 to 3 end it synthesizes the DNA, 3 to 5 end it acts as a proofread mechanism and removes these RNA primer. Now you realize that at the 3 end I'm drawing something in green. What is that exactly? Now this enzyme, you have to remember, is exclusively there only in eukaryotes. They are called telomerase. Now during DNA replication, sometimes genetic material can be lost. So in order to avoid that, we have an enzyme known as telomerase. Okay, let's go back to the list of enzymes. Now, I hope you guys have understood the DNA replication through my diagram. I tried to make it as simple as possible. Now, I can help you understand it further by revising through the list of the enzymes so that we know that we have gone through everything. So what is this origin of replication again? Origin of replication is the area where the replication begins, right? Now, you can ask me, how do we really know the origin of replication? How do we know that? Okay, so now here I have brought something known as starter box. So what is starter box? So starter boxes are 80 rich regions. Okay, they are 80 adenine and thymine rich regions. Now these rich regions are known as origin of replication. We pick these regions as origin of replication. Now replication fork. Now we already discussed that fork structure. How did the fork structure happen? Through an enzyme known as helicase. Helicase zips through the DNA, the double-stranded DNA, and forms two separate single strands that by the enzyme known as helicase. So I'm trying to draw DNA, the double-stranded DNA, but yeah, it's not being really clear. So I'm gonna try again. Yeah. So now this condensed form of DNA, I have made it into a V structure, like a fork structure. Got it? Do you recollect? So that is by the enzyme known as helicase. Now what is single binding proteins? So single stranded binding proteins. The name itself says it. On each single strand we have proteins that bind. Okay. So this helps in what? Preventing the reannealing and the degradation of each strand. Next, the topoisomerase. What is topoisomerase? Like a, topi, like a small cap. Okay, yeah. So, what does it do? It prevents the supercoiling of the DNA. Fine, supercoiling. DNA is clumped like a ball of chromatin in the nucleus, right? So we have to prevent that from occurring. Suddenly in a DNA replication, if it supercoils, then what do? We cannot perform the DNA replication, right? Like all these enzymes cannot go and bind if it supercoils. So we have the DNA topoisomerase. Okay. Next, now primase. Primase, like it says, primer. It forms a small RNA primer, okay, for the DNA polymerase to act on. See, we realize that every enzyme is helping the next enzyme to take DNA replication. That's why I put this in an order: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Fine. DNA polymerase binds to the RNA primase, the RNA primer, and carries out the replication. So basically, RNA primer facilitates the replication. Fine. I've drawn this in orange, the arrow, and also drawn here in orange, right, to show that DNA replication occurs via the DNA polymerase 3. Okay. Now you can see that DNA polymerase 3 and 1. Two things are there. DNA polymerase 3 and 1 both are in prokaryotes. Okay. Eukaryotes have another form of DNA polymerase, but the function is the same, just the name differs. Fine. Just that DNA polymerase 1 helps in degradation of RNA primer along with DNA polymerase 3, but only DNA polymerase 3 carries out replication. 3 for replication, 1 for degrading the RNA primer and replacing it with DNA. So the exonuclease part of it. Fine. Now, what next? Next, we have the DNA ligase. So what is ligase? Ligase links the DNA. So it links the Okazaki fragments, right? So we already discussed that DNA replication happens from 5 to 3 direction. So this 5 to 3 direction results in two strands, continuous and discontinuous. So these discontinuous strands, there are breaks, right? There are Okazaki breaks, fragments. So these are linked by a glue known as DNA ligase enzyme. Fine. Next, and the final enzyme, the telomerase. Now, these telomerase enzymes, remember, is strictly only and only in eukaryotes. They are only in eukaryotes. These help in avoidance in the loss of genetic material. Sometimes during DNA replication, there could be loss of genetic material. So, we add a structure, a DNA known as TTAGGG. TTAGGG. Okay, at the 3 end of the chromosome. Okay, this prevents the loss of genetic material. And we can also remember that this is mainly for, used in aging, in the process of aging. They're down regulated in aging, right? That's why the old age, they are more prone for loss of genetic material. But these enzymes also upregulated in progenitor cells and also commonly included in cancers, right? Now I have put a P. What's P? Now I'm going to bring probably an interesting fact, but do you remember Pa? P for Pa, right? So Pa movie, the child aged very quickly, right? That disease was known as progeria. That disorder was known as progeria. Now telomerase is known to be linked to progeria. Fine. Was that interesting? Do you want to know another disease that is linked to one of these DNA replication enzymes? Okay, let's go to Bloom syndrome. Now, Bloom syndrome is due to deficiency in the helicase enzyme. Helicase halves HH, right? Helicase halves the DNA, and that enzyme is deficient in this disease. Results in Bloom syndrome, the BLM gene mutation. Fine. Let's go to another interesting fact. So here is an image of a UTI infection. Okay, so you can ask me what is the relation between a DNA replication and UTI? Fascinating, right? So yeah, in prokaryotes, fluoroquinolones inhibit topoisomerase 2 and topoisomerase 4. So we remember that topoisomerase were enzymes that prevented supercoiling, right? So we can use fluoroquinolones, which is why we prescribe fluoroquinolones for UTI infection. But we never really thought that UTIs would actually inhibit something as simple as DNA replication. So, yeah, thank you so much for all your time. I'm really grateful. I hope this has made it interesting and helpful. I can't wait to get back with another lecture. Till then, bye. Take care.